Bismillahirrahmanirrahim. Assalamu alaikum. Till now, we have discussed many aspects of the hepatitis B. What is hepatitis B? What are its serology? What are the treatment targets? And what are the current available treatments? And how to treat hepatitis B in everyday clinical practice, where we translated the international guidelines into simple algorithm. In today's lecture, we shall discuss what are the upcoming therapies for hepatitis B. Bismillah ar-Rahman ar-Rahim. Topic for today's discussion is chronic hepatitis B, functional cure, and next generation therapy. Chronic hepatitis B remains one of the most difficult viral infections to cure because HBV is not just a circulating virus, it becomes a nuclear parasite. It hides inside hepatocytes as cccDNA and continuously re-establishes infection. The learning objectives for today's discussions are that by the end of this discussion, we should be able to know step-by-step HBV life cycle inside hepatocytes. We should know why HBV becomes chronic, why current therapies suppress but rarely cure, how emerging therapies target different stages of the life cycle, and why combination therapy is the future of HBV cure. Back to basics. We know when a liver becomes affected by hepatitis B and when it becomes chronic after 6 months, in the next 20 to 25 years, 5 to 30% of chronic hepatitis B patients, they become cirrhotic. And in the next 25 to 30 years after this, the 5 years risk of decompensation is 16%. And the 1 year survival is 55 to 70%, and 5 year survival is 14 to 28%. And this is because the patient develops either HCC or end-stage liver disease, and the ultimate result is death. The goal of the antiviral treatment for hepatitis B is to prevent or stop chronic inflammation initiated by HBV infection. I repeat, to prevent or stop. These are two concepts. Either you prevent the chronic inflammation or you stop the chronic inflammation, which is initiated by HBV infection. In non-cirrhotic patients, the goal is to halt the process of fibrosis and thus minimize the chances of progression to cirrhosis. While in cirrhotic patients, the goal is to halt or slow down the worsening of process of cirrhosis and to minimize the risk of development of HCC. There's another goal, that is a treatment as prevention strategy, that you reduce the pool of viremic patients and thus reduce the chances of spread to the non-infected people in the community. Remember, in HBV, we control HBV infection as defined by maintained virological response by oral drugs. And this is against HCV, where we eradicate HCV infection as defined by the SVR. And this is the main difference between treatment of HBV and treatment of HCV. And this is the main mistake which is done by the healthcare workers, that when the PCR becomes negative in hepatitis B, they stop treatment, which should not be stopped. It needs to be continued, just like treatment of diabetes, treatment of hypertension, or treatment of any lifelong disease. This is the liver and this is the hepatocytes. This is the nucleus and this is the cytoplasm. The reason I want to show this to you is that the hepatitis C, it's the infection in the cytoplasm, while the hepatitis B is the infection of nucleus. And that's the reason we can eradicate HCV, we can cure HCV, we cannot eradicate HBV or cure HBV with the current treatment. We can just control the infection, control the disease. And this is the difference between these two. We know the virology, the hepatitis B surface antigen, which has got the large surface protein. Then it has got the medium surface protein, and it has got the small surface protein. And the HBs antigen has all these three components. Then there is HBe antigen. And then there is core antigen. And in the core, we have the DNA and we have the DNA polymerase. And we know these are important to understand the phase of the infection and the serological pattern of the disease. Body makes anti-HBs against HBs antigen, anti-HB against E antigen, and then we have the core antibodies IgM in the acute phase and the total in the chronic and the HBV DNA. And these are the lab markers which we do while we detect a patient with chronic hepatitis B. Remember, HBV or hepatitis B is a lifelong disease. When a patient gets infected and the patient presents to you with acute hepatitis B, what happens? His PCR is positive, patient's HBs antigen is positive, and within the nucleus, the cccDNA is positive. We don't check cccDNA in our clinical practice, but we check these two. But remember, in acute hepatitis B, these three parameters should be remembered that these are positive. When there's a so-called cure or recovery, when patient loses the HBs antigen and the PCR becomes negative and we say the patient is cured, this is just functional cure. This is not a complete cure because the cccDNA remains still detected. It is still there. And this happens within 6 months. But we know that most of the patients, either in the acute hepatitis B becomes chronic or they are unnoticed and they present to us in chronic hepatitis B. It means the infection is there for more than 6 months. And when we treat this phase, what we do, we make the PCR negative, but HBs antigen in most of the cases remains detected, and the same is cccDNA. And this is called then partial cure. So the difference between the functional cure and the partial cure is negativity of the HBs antigen. cccDNA remains positive in the functional cure as well as in the partial cure. It is only when the cccDNA will be negative, and this we will call it complete cure. So the difference between complete cure and the functional and partial cure is negativity of cccDNA. While the difference between functional cure and partial cure is the negativity of the HBs antigen. We know there's another concept. The patient who was having the functional cure where the HBs antigen was negative, the PCR was negative, there may be conditions when the HBs antigen may become positive, and from functional cure, the patient will be transferred or will enter the partial cure. And this opens one window for treatment, and I have discussed this with you in my previous lecture. And another indication is where we treat the patient with chronic hepatitis B, where HBs antigen is positive, PCR is positive, and of course the patient is in different phases, either immune tolerant phase, immune reactive phase, immune control phase, or immune escape phase. And of course, the treatment, we need the liver status, and this is the basic determinant of treatment initiation. The patient may be having no fibrosis or minimal fibrosis, or the patient may be having significant fibrosis or cirrhosis. And these are the three main categories for treating these patients. I won't go into the detail of this. This we have already discussed in our previous lectures. This was just to link you to the previous discussion which we have done. So, to summarize the discussion so far, in partial cure, the PCR is not detected, but HBs antigen is positive and cccDNA is detected. In functional cure, PCR is negative, HBs antigen is negative, but cccDNA is still positive. While in complete cure, all the three parameters are negative. With the current available treatment, which is the nucleosides, that is the tenofovir, entecavir, and pegylated interferons, we can only achieve the partial cure. And these treatments, they suppresses HBV DNA, they normalize ALT, they prevent cirrhosis and HCC, and they achieve HBe antigen seroconversion, but they are unable to make cccDNA, which is going to be the future antiviral treatment, which will hopefully be giving us the complete cure. And in today's lecture, we will be exploring those options or those areas where the drugs can help us. Before embarking on the different treatment options in the upcoming years, the upcoming therapies, we need to know the HBV life cycle in hepatocytes. When the hepatitis B virus enters the hepatocytes and the surface antigen is left outside and the core nucleocapsid enters the cytoplasm in the form of the rcDNA, a relaxed circular DNA. And then within the nucleus, it is changed into covalently closed circular DNA or cccDNA. Then the cccDNA transcripts, different transcripts are formed, then proteins are formed, the translation, and one of that protein is the transcript is the pgRNA, which undergoes the reverse transcription, and then the RC, a relaxed circular DNA, is formed. Either it enters the recycling within the nucleus or it secretes outside of the hepatocytes to infect the surrounding hepatocytes. So this is very, very important life cycle, and you should remember this because all available coming drugs will be acting on one or the other stage of this life cycle. So the entry into cytoplasm is rcDNA, then formation of the cccDNA, then this transcription, then translation, then the pgRNA, reverse transcription, rcDNA formation either gets secreted, it secretes outside, or it enters the nucleus and initiates the another cycle. So this is the hepatocyte. This is the nucleus. And the first step is viral attachment and entry. This is the hepatitis B virion, and the NTCP receptor, it gets attached to it. Then there's endocytosis, and the surface antigen remains outside and the nucleocapsid enters. So HBV specifically targets hepatocytes because of the NTCP receptor, this one. The pre-S1 domain of HBs antigen binds anti-receptor and the virus enters via endocytosis. So this is the first step of the life cycle of hepatitis B virion within the hepatocytes. So this step allows a therapeutic target. And what is that? Bulevirtide, the entry inhibitor. It blocks entry alone, but remember, the blocking entry alone cannot cure established infection because infected hepatocytes already contain cccDNA. So it will prevent the entry of the other virions, but whatever cccDNA has formed within the nucleus, it cannot affect that. So the entry inhibitor, especially, it is important to prevent the new hepatocytes to get infected, but the infected hepatocytes is not cured. So, but the step one is the treatment opportunity for the bulevirtide, and now it has been FDA approved for the hepatitis D and hepatitis B as well. In the Europe, it was approved by the European Medical Authority for hepatitis D, and now for hepatitis B, it has also been approved. Then the second step is this one. That was the first step, viral attachment and entry. The second step is uncoating and nuclear transport. The capsid, it uncoats, and the nuclear transport to the nucleus. So after entry, envelope is removed, nucleocapsid travels towards nucleus, relaxed circular DNA enters the nucleus. So the virion enters the nucleus as relaxed circular DNA. HBV is real home, is not blood. Remember, it is the hepatocyte nucleus. It resides there. So this is another potential site for drugs, but till now, no drugs approved for this. There are in phase 1, phase 2 trials are being conducted, but not in phase 3 trial. Any drug is here which can affect this uncoating and nuclear transport. For the first step, the bulevirtide has been approved, but there is no drug approved yet for this phase. Then comes the third step, that is the formation of the cccDNA from relaxed circular DNA. This is the most important step in the entire HBV life cycle. Relaxed circular DNA is repaired by host enzymes into cccDNA, which means covalently closed circular DNA. And remember, the cccDNA behaves like mini chromosome. It's a stable viral archive, and it's a permanent transcription template within the nucleus. So it behaves these three ways: minimal chromosome, stable viral archive, and permanent transcription template. And the cccDNA, I must say, is the Achilles heel of HBV cure research. So the drugs in process for this step are direct cccDNA targeting therapies, just like direct antiviral therapy for hepatitis C. And these include inhibitors of cccDNA formation from RC to cccDNA. Then there's direct cccDNA destabilizers or eliminators. Whatever cccDNA is formed, they destabilize it and they remove it from the nucleus. Then the third group is gene editing therapies. And the fourth group is epigenetic silencing of cccDNA. So these are the four different classes of direct cccDNA targeting therapies: inhibitors of cccDNA formation, Then if the cccDNA is formed, direct destabilizers or eliminators, then gene editing therapies and epigenetic silencing of cccDNA. And the examples of these drugs are gene editing, CRISPR, Cas9, talin, and ZFN, and epigenetic silencing of cccDNA. So for the third step, the most promising therapies are here and they are in different phases of research, and hopefully in the next 3 or 4 years, we will be having these drugs available for us as we are having drugs available for hepatitis C direct antiviral therapies. Then the fourth step is from this rccDNA transcription. The cccDNA uses the host RNA polymerase 2, and its transcripts are produced. Two types of transcripts are produced. One is pre-genomic RNA and sub is subgenomic RNA, messenger RNA. So pre-genomic RNA is one transcript and there are subgenomic messenger RNAs. So these two types of transcripts are produced by the cccDNA using the RNA host RNA polymerase type 2. So the next class of drugs are targeting this step 4, and these are the RNA targeting therapies. These are the small interfering RNAs and antisense oligonucleotides. These two are the classes of drugs which will attack the hepatitis B virion cycle at the step number 4. And examples of these are bepirovirsen and imdosiran. These are the available drugs and they are under the research. And the effect of these will be that they will reduce the HBs antigen production and they will allow immunity recovery. Then is the fifth step, and that is the translation of viral proteins. These two transcripts are formed and know how these are translated. After transcription from cccDNA inside the nucleus, pgRNA and sgRNAs move into the cytoplasm, where they are translated into viral proteins by host ribosomes. From pgRNA, the HB core antigen protein is formed and virion polymerase or P proteins are formed. So two proteins are formed from pgRNA, the HB core antigen, and viral polymerase, which is called P protein and core protein respectively. And from the subgenomic RNAs, surface proteins, that is the HBs antigen, are formed, HBe antigen is formed, and HBx protein, which is the regulatory proteins, these are formed. So 5 types of proteins are formed from these transcripts from pgRNA, core antigen, and viral polymerase, and from subgenomic RNAs, surface proteins, E antigen, and HBx protein. So the summary of these from pgRNA, HB core antigen, and polymerase, and their main function is replication and capsid formation. Whereas from the subgenomic RNAs, HBs antigen is proteins are formed which make the envelope formation, then E antigen, which is important for immune tolerance, and HBx protein, which is important for replication and carcinogenesis. But just like the second step, for the fifth step as well, no drugs are approved yet. Then the sixth step is nucleocapsid assembly. Encapsidation, which means HBV replicates like a retrovirus. The packaging, the core proteins assemble around pgRNA plus viral polymerase, and the immature nucleocapsid is formed. So the capsid assembly modulators, this is another group of drugs which is under development, and they will be also hopefully available to us in the next 5 to 7 years. And these mechanism of these drug is that they disrupt nucleocapsid formation, and they block pgRNA packaging, and they reduce viral replication. And the effect of these drug is they inhibit viral replication cycle at core level. And the examples of these CAMs or capsid assembly modulators are AB423, GLS4, and JNJ6379. And these molecules, these drugs will hopefully be available for clinical use in next 5 to 7 years. Then is the seventh step, and that is the reverse transcription. HBV replicates like a retrovirus, I already said. So inside the nucleocapsid, the pgRNA makes negative strand DNA, then the positive strand DNA, and it is then the relaxed circular DNA. The current standard therapies, that is the tenofovir and entecavir, act here at the reverse transcription here. This is the site for the available drugs. So the nucleotide and nucleosides, the high barrier drugs, these are the tenofovir, TDF-RTF, entecavir, or bisulfovir. And then others are telbivudine and the lamivudine. Then in the eighth step, there are two onward pathways. Pathway A, recycling, as I mentioned to you while I was describing the life cycle, that the 90% of the mature capsid traffic back to nucleus. Import relaxed circular DNA to replenish the cccDNA pool, and this amplifies infection without new virus entry. So the inside recycling is done. And the pathway number 2 is, or B is, envelopment. This was recycling, this is envelopment. The mature capsid dock with the HBs antigen in endoplasmic reticulum and Golgi, and the envelope is acquired complete virion but into endoplasmic reticulum lumen. For this step, still no drugs approved yet, but maybe in the 7 to 10 years, drugs may come on this side as well. Then the ninth step is release into blood. The HBs particles exit to blood via multivesicular bodies, and these contains DAN particles, which is the infectious HBs antigen particles, and subviral particles, which are the non-infectious HBs antigen particles. These are the empty HBN spheres and filaments. So the HBs particles, and they release into blood in these two forms. The subviral are non-infectious particles. They are made in 1,000 to 100,000 excess. They are non-infectious, but they act as immune decoys by soaking up anti-HBs antibodies made by the host. So they overwhelm the immune system, they maintain tolerance and contribute to chronicity. And this is the reason that the virus becomes chronic. Yes, inhibitors of antigen secretion, they are in the research, and these are the nucleic acid polymers, for example, REP2139, REP2165, which blocks HBs antigen release. And then the HBs antigen assembly inhibitors. And the importance of these are that they reduce antigen burden and they restore immune response. And they can produce large HBs antigen decline up to 3 log. This step. And the 10th step is integration. The HBV DNA may integrate into hepatocyte genome, and the consequences are persistent HBs antigen production, genomic instability, and hepatocellular carcinoma. Important point here is that even when replication is suppressed, integrated HBV DNA may continue producing HBs antigen. And for this step again, no drugs approved yet. Then the immune dysfunction in HBV, this concept. Chronic HBV is not merely viral persistence. It is also immune failure. The features of this immune dysfunction is there's a T cell exhaustion, defective innate immunity, and high antigen burden. So the HBV cure requires both virological control and immune restoration. And this is the point you should remember, that if you want to cure HBV, you need HBV virological control as well as immune restoration. So for this purpose, there are innate immunity stimulators like TLR agonists, then REG1 agonists, and there's adaptive immune strategies, therapeutic vaccines, and checkpoint inhibitors, PD-1, PD-L1 blockade, and engineered T-cell, CAR-T or TCR therapies. So these are the drugs which will be the immune modulatory therapies, and they will restore the immune status of the body. So the combination therapy, the concept to combine is viral suppression with antigen reduction and cccDNA targeting and immune restoration. So the combination therapy is going to be the future of the curing of hepatitis B virus infection, where you will suppress the viral replication, the antigen reduction, the cccDNA targeting, as well as immune restoration will be required. And the evidence strongly suggests that single drugs is not going to give you the cure. This is the combination approach, which is the best chance for functional and complete cure. And the example of the combination will be nucleoside analog plus small interfering RNAs, the Na plus siRNA plus immune therapy, nucleoside analogs plus capsid immune acting molecules and immune therapies, plus Na pig interferon plus vaccine, and multi-drug regimes. So this is the better you take a screenshot of this. This is the life cycle of hepatitis B virus. It's the steps, the attachment and entry, then uncoating and nuclear transport. This one, then the formation of the cccDNA, then transcription, then translation, nucleic capsid assembly, reverse transcription, two pathways, and then this is the release, and this is the integration. So these are the different steps which has been shown here, and these are the available treatment options for each step, what options are available and what are not available yet, and this is the immune modulation. So for complete or sterilizing cure goal, we need elimination of the cccDNA plus integrated HBV DNA plus restoration of HBV specific immune response. So this diagram, it summarizes the whole presentation which I did with you, and I will suggest that you take the screenshot of this with you and keep it with you, because this is going to be the main template for the upcoming therapies for hepatitis B cure. So the key takeaways from today's discussion is that the story of HBV is no longer just about suppressing a virus. The story of HBV is no longer just about suppressing a virus. It is about dismantling a viral ecosystem hidden inside the hepatocyte's nucleus. Thank you very much. I end here. If there are any queries, any suggestions, any points, you may kindly share those in the comment section below, and I will try to answer those queries. Thank you very much. See you in the next lecture on some other topic. Till then, take care. Khuda Hafiz.